Dr. Huentelman is part of Tgen, and they're taking a little bit of a different approach to MSA. They're looking at some of the genetics, and he's going to explain what their study entails.
It's really wonderful to be here. We imposed a very last-minute request on the organizers, and they were kind enough to honor it. And so I really, really appreciate that. We are brand new to the field of MSA, but we are not new to human genetics. So we're very excited to apply our approaches that we've used for other rare disorders to MSA and start to explore that new piece of intel. And is there a role for genetics in this disease? Secondly, I just want to say the talks that you guys heard today, especially the talks this morning, focused on explaining a very complex disease, very complex physiological processes in very simple terms, were just fabulous. I've been to a lot of meetings like this where physicians and scientists collide with patients and caregivers, and those were some of the best I've ever heard. So you guys, me as well as you, are walking away with some really great knowledge base today. And lastly, I just want to say that, scientists, so I'm a PhD scientist. We think that on Sunday morning we can wake up and make a cup of coffee and read the literature, and I can learn all about MSA by reading the scientific and medical literature, okay? I'm here today to guarantee to you that's wrong, but that's the point that I'm at right now, but that's why I'm standing here today, is to learn from my colleagues in medicine and research, but also learn from the patients and caregivers about where should we put our time and energy for this disease. Because this is really, meetings like this is where we really get down to the heart of the issue and what we should focus our efforts on. I can't glean that by reading the scientific literature or the medical literature from my kitchen at home, okay? So I really, really appreciate the chance to be here. And it has been fabulous. So we've come a long way since the very first pencil drawing of DNA. This is a drawing by Crick 50 years ago. But I have to start the story here with Shelby Valent. Shelby Valent is my motivation for studying the genetics of rare disease, and it's one of the reasons why I'm interested now in adult rare disorders. Shelby suffered from a rare childhood disease. Interestingly enough, it ended up to turn out we identified that it was a dopamine disorder. But we only found that out by sequencing her genome, her genetic blueprint. We'll talk a little bit about what that means. And I'll try and introduce that whole concept of DNA and RNA and proteins to you guys, hopefully as good as these guys did the very complex physiological systems this morning. But we only identified the cause of her disease after she was living with it for 10.5 years by sequencing her blueprint. Shelby was wheelchair bound, had a hard time eating and speaking, but as soon as her physician now knew what the core cause of her disorder was, a dopamine regimen got her out of her wheelchair. This is a picture of her dress shopping with her mother for the first time in her life. So that's her in the fitting room with her new dress on. But she's the motivation because we've seen such an improvement in the diagnosis of these rare childhood disorders. I'm not trying for a minute to equate MSA or the other adult neurological disorders that are rare to our childhood disorders, but this is my motivation. We've seen a big increase in our ability to diagnose these, about 8 to 10 times increase in diagnostic rate in kids with rare neurological disease using genome sequencing. That's in our hands with actual data, sequencing about 50 families now. So that's the motivation. I'm coming to you from Tgen. This is a picture. When you hear speakers from universities, they don't have to introduce themselves or their institute, but I have to because everyone wonders, what is Tgen? So Tgen is a nonprofit research institute. It's short for Translational Genomics Research Institute, so we'll stick with Tgen for the rest of the talk. We are nonprofit, so we're sort of like a Salk Institute, Scripps Institute. You might have heard those names before. Phoenix, Arizona, and we're only 10 years old, so we're relatively new kids on the block. but that's because we focus on the human genome, and it's only been about 10 years since we've been able to actually fully understand the full breadth of knowledge that is the human genome. So the first genome was really finished sequencing about 10 or 12 years ago. We have a focus on brain disease, or neurological disease, oncological, metabolic, and microbial-based diseases, but I'm here today because I study brain disease, or neurological disorders more accurately. We have a significant technological base in genetics, and genetics now uses massive supercomputing resources, so that's another area of expertise. And we focus pretty heavily on translating our work, so doing science that actually makes a difference for the patients today and hopefully helps us avoid disease in the future. That's really our core mission. Okay, so this is my attempt to introduce you guys to my complex topic, okay? So this is how I like to think about and have you guys think about the role of your genome, your genetic blueprint in the cell and in your bodies. So I like to think about it as if it were a construction site. And if you want to imagine that your DNA, your blueprint, is sitting there in the hands of the foreman in that protected, so in Phoenix we do construction year-round, but at this time of the year, man, it's hot. So you better have an air-conditioned place to pore over those blueprints. So if you imagine that the foreman's looking at that DNA, that blueprint, inside that air-conditioned trailer, that's sort of like the nucleus of your cell. It protects that DNA. Imagine in the morning the foreman meets with project managers. So we have people who have to do the plumbing and people who are laying electrical line. They come in the morning and they get their marching orders, if you will. And it's a small number of people, and then they go outside the trailer and they take that message out to their workers, and they have big teams of people who are now going to follow those instructions and put down that electrical line for the day and lay the plumbing for the day. So if you think about the process of a construction site in that way, that's sort of how your cells work, with DNA being the blueprint. RNA is like a sister chemical of DNA. That's sort of like the project managers. RNA is made in your nucleus, and you can go outside of the nucleus, travel outside into the cell, and it can be formed into proteins. Just for the sake of argument today, proteins do stuff. Okay, they're just like the worker bees out there laying the electrical wire and the plumbing, etc. But there's a missing part to the building, and that's your environment. In the case of construction, it's your raw materials. What kind of grade of electrical wire did you buy? Who'd you buy it from? That's all really important as well, the quality of those materials. So all this adds up to a well-built building or a functional building. But if you think about this now from the context of your body and from how disease can happen, hopefully you can understand how breakdowns at any one of this point can lead to bad electrical wiring or subpar plumbing. And that can manifest as disease, okay? You can also hopefully appreciate that it can be the combination of problems with the blueprint and the bad supplier of your cement that can lead to disease. Does this make sense to you guys? So it's this sort of combination of things that really plague most of the disorders we're focused on today. My life changed 5 years ago. So did all the geneticists across the world, because we finally moved to the digital age. The very first human genome was sequenced on analog devices, if you guys can believe it. Analog devices by hundreds of bodies and thousands of machines. Now we finally use digital machinery to generate our data, and that's changed everything because it's reproducible, it's accurate. Day to day I get the same results. I can do it quickly. I can do it more efficiently. So the first genome took about 10 years and about $3 billion to do. We now do the genome overnight on these machines. It is really amazing, and that's the progress in just 10 years. It no longer costs $3 billion either, or I wouldn't be here talking to you guys today. So it's actually affordable now today to do it, in the thousands of dollars to do a genome overnight. So that's the pace of genetics research, and that's why we're excited, because this is a brand new tool for for most diseases out there. So while traditionally MSA has not necessarily been thought of as a genetic-based disorder, this is a new tool now to ask that question more thoroughly than we ever could before. So we don't want to ignore it. In my opinion, we shouldn't ignore it. We should give it a shot. And because I can now sequence the genome, it's sort of like moving from the Cliff's Notes version to actually reading the whole novel. I don't know about you guys, but that's how I survived college. But I'm now smart enough, I think, to appreciate the fact that reading the whole novel would have actually been a much more deeper experience for me. And that's what we can finally do. This is the obligatory cost graph that I must show you as mandated by the NIH. I'm kidding, but every geneticist shows this. This is the massive drop in genome sequencing costs. This is why the genome is looming for our healthcare. It's going to collide with our healthcare in the very near future because we're looking at getting down to the costs of, let's say, an MRI. The same cost of an MRI to sequence your genome. How can we ignore that? Well, it's coming, I think. Okay, but the data sets are massive. That's why we have to have our computational technologies all in line, and we have to be ready for this deluge of data. So your genome is 3 billion letters long. Nobody knows what 3 billion letters is, except we know it's smaller than the debt. Yeah, which is scary. After I give you an analogy here, you'll be even more scared about the debt. So if I printed off your genome, 3 billion letters, standard sized notebook page, standard sized font, no tricks here, and we stacked it up, it would be as tall as the Statue of Liberty. Okay, 300 feet tall for 3 billion letters. But scientists are masochists, so we don't trust ever reading a novel one time. So I would never want to read your story once and have an error. I like to read it 30 times, which is why I used an old photograph here, because there are the Twin Towers. So if I read your genome 30 times and print it out, it's about 6 times as high as one of the Twin Towers. So, massive amount of data. Thankfully, we have spent, since the inception of Tgen, we've spent all of our effort focused on interpreting that and reducing this down to lists that we can make sense of and actually test hypotheses. Okay, any questions from that first bit? Just a few more slides and I'm finished. Okay, so I think we can probably all agree that MSA is a complex human disease. To me, that means it's probably the combination of genetic risk factors and environment. And unfortunately today, we just don't have the answers for either of those parts of the equation. Very few diseases are 100% environment. And that's why I'm compelled to take a look at the genetics of MSA, even though traditionally it's been thought of to be non-genetic. The example I give about a disease not ever being 100% environmental is the example of HIV. So HIV is caused by a virus, a viral infection, right? Matt, how can it have anything to do with genetics? Well, there are actually, the way HIV enters cells is it uses things on the outside of the cells called receptors. This is a cartoon of one of the receptors for HIV. Some of us in the room carry a form of this receptor that's called delta 32. And that actually makes it harder on the virus to get into our cells. And very few of us have 2 copies of that deletion in that receptor, and that really makes it hard for HIV to infect us. So even for a disease which is really rooted 100% in environment, there's still some clues that we've learned along the way of studying HIV that have been 100% genetic. And of course, this led to the development of some drugs targeting this exact receptor. So how do you get to a rare disease, though, with genetics? And this is just me thinking out loud in front of all of you. Let's consider just a few examples. So here's an example of a rare, what I call a rare disease, PKU, and it's caused by rare mutations in a gene called PAH. This is just going to be a theme. You don't have to memorize what I'm saying here. But the environmental cause of PKU is actually common. The environmental cause is intaking dietary phenylalanine. Okay, that's in pretty much any protein source you eat has phenylalanine. Okay, so we have a rare genetic cause, a common environmental insult. Think back to the building example that I gave you, but causing a rare disease. What about type 2 diabetes? That's the abbreviation T2D. That's a common disease, and it's known, there's a known common genetic risk factor called TCF7L2. Don't worry about it. But it's common. The environmental factors that send you down the path for type 2 diabetes are also common, unfortunately. If you make poor choices about diet and exercise, you increase your risk for type 2 diabetes. So that results in the commonness of type 2 diabetes. I might have the most extreme risk genetically for type 2 diabetes, but maybe if I make the right diet and exercise lifestyle choices, I never get the disease. Okay? So genetics is just chemistry, it's not destiny, but so is your environment as well. Okay? So then you have to start to think about MSA. So could we get to the rare disease of MSA through rare genetics and common environmental experiences? Yes, I think we could. Could we get there through rare genetics and something rare in the environment? Yes, we could, I think. And could we get there through common genetics and something very rare in the environment? Yeah, we probably could as well, but I think we would have already explored this common genetic link. But nonetheless, so this is sort of how we're thinking about this. And again, just thinking out loud with you guys here, but hopefully that makes sense. So, last couple slides here. So, for me, when I approach this disease, and again, I'm looking forward to collaborating with clinical partners, these guys and others across the country, to better understand some of their actual clinical experiences with patients with disease. Because some of these things that I can read in the literature are actually very interesting to me. So you're probably well aware of the Japanese study that looked at families that were segregating MSA. That means 2 people in the family had the disease. That's, of course, suggestive of genetics. And they recently reported on the genetic change that they found that was a risk factor in those families. Unfortunately, that did not translate to Caucasians in the US. That was not found to be a risk factor in their study for Caucasians in the US. But that showed that there could be genetic risk factors. And so you either have to believe that that's only for the Japanese population, or could there be risk factors outside of Japan that we haven't found yet? I don't know. The subtypes, to me, as a geneticist, are really interesting, and further trying to classify them into other subtypes is also really, really important, because the more cleaner subtypes we can investigate, the better powered we are to find the genetic cause. To me, it's warning bells to combine MSAP and MSAC and try and study them together. That can really cloud the results. So it's motivating to look at these 2 subclasses separately. And the heterogeneity, clinical heterogeneity, the disease duration. I've read reports of people living for long periods of time, let's say 15 years. That may be an absolute rarity in you guys' opinion. And we should talk more about that, because I think those patients that do reach, let's say, 15 years, to me, I want to know if they have a protective factor in their genome that's extending that disease duration, because that's a great drug target if we can find it. And the levodopa responsiveness. So we've heard a lot about dopamine today, and there are reports that some MSA patients actually have at least a small or partial response to L-DOPA. So do they have an entirely different disease altogether, or is there something interesting about their genetics that's eliciting that partial response? Okay? So we've started down this path, and what we've done is we've sequenced the entire genome of 2 families. Families are powerful for us because we can use all the information from mom and dad to subtract out the genetics of the child that has the disease. So these are the 2 families. This is, circles are, well, squares are, of course, guys, okay? Where are the squares in this? Okay, that's an easy one. And this is showing 3 siblings here, 1 with disease. Over here, 2 siblings, one with disease. One of these was an MSAP and one was an MSAC. And interestingly enough, we found some initial findings in these families. They need to be extended. We need to do much larger studies. So I don't really even want to go into the findings because I think it's way too premature. But I'm here today to tell you guys about our research trial, which we'd like to open. We'd like to open it because it's not ready yet, but we've created a registry. And so the registry website, and I have handouts for you guys to take with you. I haven't passed them around, but I don't know why. When they made this, they stuck my face on it. I guess you'll remember who gave you this talk today. But at curemsanow.org, which is really a site within Tgen, you can just simply stand up and raise your hand and say, I'm interested in being a part of this study when you're ready to contact me. And we ask a few questions about your disease. And also, most importantly, we ask about who's your treating physician. Tgen doesn't run neurology clinics. We partner with physicians to better understand their patients. So we're interested in this being an absolute partnership with your treating physician, you and us side by side, trying to understand the genetics of MSA. So if you're interested, you can go there. Like I said, it's just it's a registry, so you're just sort of raising your hand and saying, Matt, when you're ready to launch the study, let me know with a simple email, and then we'll start enrolling. So why is this important, Matt? Why are you making me do this now? It's important for us when we go to seek funding, be it from federal, from the NIH, or from people who might just be interested in supporting this, that we can show there's an interest out there in the community. So this is a demonstration that people are interested, they're willing to participate. We've been able to create a registry. It can also be valuable for others performing research in this field as well. So I'll leave you with a shot of a forest that unfortunately burned this last year, but it sure was pretty, I guess. So thanks.